Grandma, why are you so weak? After all these years, why does your body decide not to work anymore?
Sweetie, deep in the cells of my body and my DNA, certain genes are turning off like a light switch.
But what kinds of genes are turning off, and where exactly?
Well, honey, I was just reading this article about that.
Our article is Epigenome-Wide Scans Identify Differentially Methylated Regions for Age and Age-Related Phenotypes in a Healthy Aging Population. First of all, methylation is catalyzed by enzymes involved in regulation of gene expression. It is the addition of a methyl group to a cytosine pair. DNA methylation is an epigenetic mechanism that plays an important role in gene expression regulation, development, and disease. The genetic environmental and stochastic factors contribute to DNA methylation levels at individual genomic regions. Specific DNA methylation at these sites vary over time within an individual, and age-related methylation change have been identified in multiple tissues and organisms. Aging is a catalyst of degenerative processes. Figure 1 shows the two main components of the epigenetic code, DNA methylation and histone modification. Figure 2 shows the methylation process of methyltransferase.
Biomarkers and age-related phenotypes. Biomarkers are defined as occurrences in cells and molecules that alter future events. There are specifically 16 phenotypes, some including blood pressure and lung function. Biomarkers can also affect maternal and paternal longevity, as well as age at reproduction. During this experiment, researchers studied different types of methylated regions in a sample of 172 female twins and compared DNA methylation patterns with chronological age. Then they related epigenetic variation to age-related phenotypes that have previously been used as biomarkers of aging. Epigenomic-wide association scans. This type of scan is used to identify differentially methylated regions for age and age-related phenotypes in a healthy aging population. Human degenerative processes. Aging is a catalyst of degenerative processes. Some of these processes include oxidative stress and DNA repair. To conclude, there seems to be a correlation between these processes and methylation as phenotypes are altered.
The scientists at PLOS Genetics compared DNA methylation patterns to age and age-related phenotypes to gain insight into potential mechanisms underlying age-related DMRs. Basically, they were able to use technology to determine the exact cytosine-guanine pair where methylation was occurring, and then they related that location on the DNA back to what gene it was. Differentially methylated regions, or DMRs, associated with age were identified as those that surpassed the 5% FDR threshold. These graphs are epigenome-wide association scans of age and age-related phenotypes. These scans identified age-related DMRs associated with low cholesterol level on the first graph, lung function on the second graph, and maternal longevity on the last two graphs. In contrast, the scans for chronological age identified hundreds of predominantly hypermethylated age DMRs, of which only one was associated with the age-related phenotype, and that was the maternal longevity, which is represented on the far right graph.
So, in conclusion, the majority of changes related to age are not associated with changes in the environment. In fact, some of these genes may be inherited. These scientists concluded in their paper that in a small proportion of genes, DNA methylation may be used for gene expression of age-related phenotypes.
DNA methylation patterns were studied in monozygotic twins or identical twins, dizygotic twins or fraternal twins, and unrelated individuals. In order to characterize DNA methylation patterns, 26,690 promoter CpG sites of each of 172 middle-aged female twins were analyzed. Methylated regions and unmethylated regions were identified by treating a microarray of DNA with bisulfite. This treatment caused the unmethylated cytosine regions to be converted to a thymine nucleotide. The methylated regions were protected by the methylated cytosine nucleotides and were not converted to the thymine nucleotide. Fluorescent signals were then detected to determine the relative level of methylation. This is known as a beta coefficient, and it represents the methylated regions to the methylated regions plus the unmethylated regions. The research group observed that the majority of the autosomal CpG sites were unmethylated, with a beta coefficient of less than 0.3. They also observed that the X chromosome CpG sites were hemimethylated, and hemimethylated means that the methylation was detected on only one strand of DNA. The group found that monozygotic twins had more similar DNA methylation patterns than the dizygotic twins, and that the twins had more similar DNA methylation patterns than the unrelated individuals. Correlation coefficients were obtained that describe the degree of relationship between the two variables, relatedness and DNA methylation frequency. The correlation coefficient obtained for monozygotic twins was the greatest, and the correlation coefficient obtained for unrelated individuals was the lowest. This indicated that DNA methylation is heritable.
DNA methylation has been known to show patterns in the cells through the processes of mitosis and meiosis. As we already know, mitosis and meiosis are processes of cellular replication, so the DNA within these cells are either genetically identical, such as in mitosis, and genetically very similar, such as in meiosis. So the patterns of methylation can then be developed in similar patterns in the daughter cells due to the inheritance of DNA traits. We also know that a person's phenotype is the outward expression of a gene. Due to methylation studies, there are now links between biological age, which is calculated through hereditary and environmental factors, and the aged expression of certain phenotypes. This explains why the elderly are much often weaker, have less dense bone structure, and have a high susceptibility to disease, such as grandma's weak bones in this case. As age-related phenotypes actually age, and as seen through epigenetics, which is the study of gene changes, the cell structure begins to deteriorate, causing a change in the patterns in DNA replication, which could be in part to enzymes such as DNA polymerase, which make copies of one strand using DNA templates, are denaturing. Simply put, we as humans cannot limit ourselves to a chronological age, since our internal processes have a more complex structure behind phenotypes than we actually realize.
That is really cool, Grandma. I think I want to study genetics when I grow up.
I think that'll be a really interesting job.